Okay, this is about multiple alleles. In all examples given so far, there have existed only two alleles for each trait. Example, yellow or green seeds, red color or white, attached earlobes or free earlobe. In some cases, however, there exist in the population more than two alleles for a given trait, or multiple alleles. For such traits, it is important to understand that although more than two alleles exist in the entire population of an organism, each individual organism can still only have two of the existing alleles, as always, one from each parent. For us, we're going to need to know these symbols. So capital I represents all dominant alleles. There's more than one dominant allele. Little I represents a recessive allele. There's only one recessive allele. We're going to use the example of human blood types for this. Human blood types are the result of certain proteins embedded in the cell membranes of red blood cells. There are three alleles that code for these proteins. I, capital A, is the dominant A allele. It codes for red blood cells protein A, or antigen A. I, capital B, is dominant B allele. It codes for the red blood cell with protein B, antigen B. And little I is a recessive allele, codes for absence of red blood cell proteins, no antigen. The alleles for red blood cells proteins A and B are codominant. This means that when both alleles are present, they are both fully expressed. The proteins that are embedded in the membranes of red blood cells are called antigens. In addition to red blood cells, human blood plasma, the liquid part of your blood, also contains proteins called antibodies, whose job is to recognize and help destroy foreign cells. When antibodies encounter a foreign antigen, a foreign red blood cell protein marker, they combine with it, causing red blood cells to clump. And you don't want your blood to clump, because that's fatal. So here's the status of how we look at the blood types. You have genotype I, capital A, that's homozygous, or I, capital A, little I, that's heterozygous. In the blood, there's red blood cells have protein A or antigen A on their surfaces, and there's anti-B antibodies in the plasma. Your phenotype would be a type A blood. If you were exposed to anti-A antibodies, your blood would clump, and if you're exposed to anti-B antibodies, your blood wouldn't clump. Then you have capital I, A, IB, that's homozygous, and capital I, little b, I, and that would be anti-A antibodies would be in your plasma. You'd have protein B on the surface of your cell membranes and of your red blood cells, and your phenotype would be type B, and you'd have clumping if you were exposed to anti-B antibodies. And then there's IA, IB, and that's a codominant situation. That means you have no antibodies for blood protein, nothing going on there in the plasma, but you have both A and B antigens on your red blood cells. You're type AB, and if you're exposed to any antibodies whatsoever, your blood would clump. Okay, then it's on to little I, that's, you have both A and B antibodies in your blood, but you have no antigens on your cell membranes of your red blood cells. Your type O is a phenotype, and you'd have no clumping if you're exposed to antibodies, since you already have the two antibodies in your bloodstream. And this is just the same thing. Okay, so the most common blood type out there in our population is O positive, then O negative. The most rare is AB negative. The universal donor, you can give blood to anybody, is O negative. And the universal acceptor, you can accept blood from anybody, is AB positive. But it's very tricky. You have to make sure that you're getting the right blood. As you can see, you don't want to have your blood clump, and that's no good. Okay, let's look at the inheritance factor about this. So a woman who is heterozygous for blood type B marries a man who is heterozygous for blood type A. Use a Punnett square to determine the possible blood types of their children. All right, so we have heterozygous type B, heterozygous type A. Let's set it up. Let's get the Punnett going. And that's how you would set up the Punnett square. So we're going to have about a quarter of them could be type A, 25%, 1 out of 4. 25% could be type B, 25% are type AB, and 25% are type O. Sheila and Tom are married. Sheila, who is heterozygous blood type B, has a daughter, Katie, who is heterozygous blood type A. When Katie developed a serious genetic illness, it became critical that her complete hereditary background be determined. 3 other men came forward, all claiming to be Katie's biological father. We got Tom, that's type B. We got Joe, the milkman, that's type AB. Sean is a type O, and Tony, the postman's type B. Obviously, it's Joe, the milkman, because he's got an A allele, and nobody else does. Okay, sex-linked inheritance, a pattern heredity in which traits are transmitted by genes located on the 23rd pair or the sex chromosomes. That's called sex-linked inheritance. Traits that are determined by genes located on the X chromosome are said to be X-linked traits. Sex-linked inheritance was first observed by T.H. Morgan in the early 1900s at Columbia University. Morgan's research provided final proof that genes are carried on chromosomes because he could trace a specific trait to a specific chromosome. Morgan used the fruit fly, the Drosophila melanogaster, as his test organism. What are some reasons why Drosophila is very well suited to genetic research? Fruit flies have a short life cycle, 2 weeks. They produce large numbers of offspring. Fruit flies need very little food and space. Fruit flies have only 8 large chromosomes or 4 pairs. Fruit flies have several easily observable physical traits, and you can really tell the difference between male and female fruit flies. Why are some traits carried only on the X chromosome and not on the Y chromosome? Well, remember that the Y chromosome is much smaller than the X chromosome. It only carries actually the genes that make a male. It just carries genes that code for male secondary sex characteristics. It doesn't have anything, I don't think so. I don't think it has anything that's connected to the X chromosome. So let's look at the fruit flies. Eye color is a sex-linked trait. The gene for eye color is carried on the X chromosome. Their wild type color is like a reddish brownish color. The allele for red eyes is capital R. It's dominant to the allele for white eyes. White eyes is actually a mutation caused by something that interferes with the way that the pigments move to the eye cells. There's a transport protein that's not being produced, and therefore it's not going to, the little pigments are being made, but there's no truck to take it over to the eye cells. So that's the issue there. So anyway, back to this. So eye color is sex-linked. We set up this situation. So if you see XX, you know it's a female. If there's capital R, that's homozygous, and that would be red because red is dominant over white. Then you have female here also. See, now you have to designate female and male on this one because it's sex-linked. And then this would be heterozygous, and this would be a red-eyed female. This is a female, and this is also homozygous, or this is homozygous, and this would be white-eyed, which is very rare, but that can happen. And now we'll look at the males. So this is a male fly, and it's neither because Y is not like another X, so there's no homozygous, heterozygous. It's just neither. And this would be a red-eyed male fruit fly because of the capital R there. And then let's do another male here, and this would be neither, and that would be a white-eyed fly because it carries the recessive form of the trait, or the recessive allele of the trait on the X chromosome. So what are the results of a cross between a heterozygous red-eyed female with a red-eyed male? Okay, so let's see. X capital R, X little r, X capital R, Y. So male and female. Now we'll put it in the punnet like this. This is how you set it up. And now you're going to find out that we have to separate male from female since this is sex-linked. Okay, so you have X capital R, X capital R, X capital R, X little r. So that means 100% of the females, 2 out of 2 boxes, is red-eye. X capital R, Y, X little r, Y. That means half, 50%, 1 out of 2 males here are red-eye, and 50% are white-eyed. Will be, could be. This is probability, of course. So you have to do it with the male and then female, and that's how you look at it. That's how you're looking at it. Okay, what are the results of a cross between a white-eyed female and a red-eyed male? All right, let's set that up. Okay, so what we see is there's 100% red-eyed females again. These are both red-eyed, 2 out of 2. And no red-eyed males, but they pretty much all the males would be white-eyed in this next generation, according to this Punnett square, because these are both X little r, Y, X little r, Y, so they're all white-eyed. Now, in humans, hemophilia, bleeder's disease, is a sex-linked trait. It is a recessive genetic disorder caused by a mutation on the X chromosome. This mutation causes the body to build a defective form of a protein needed to clot blood. This results in reduced ability to clot or seal and heal wounds. And they have this thing where it's been mostly males, of course, that get hemophilia. And they looked at Queen Victoria's male descendants, and they all said that they were sick and they had bad health, and we pretty much just figured out that it was because of hemophilia passing through their line. It was a world disease spread through heirs to thrones across Europe, including Britain, Germany, Russia, and Spain. When solving problems involving human sex-linked disorders, biologists often use the letter of the disorder to represent the defective allele, even if it's recessive. In this case, we would use little h to represent the allele for hemophilia. Color blindness is also a sex-linked trait, and that would be represented by a little c, representing the recessive form. Females who are heterozygous for the trait don't show the traits themselves. They won't be having that actual disease, but they can pass it on to their offspring, and they're said to be carriers. So let's take a look at this situation, similar to little flies. You have the male and the female designated out like this. Okay, so then you have XX, female, homozygous, normal. You have X with an X little h on the top, female heterozygous, that's a carrier. That female will not be expressing hemophilia. She will not be hemophiliac, but she can, because she's got the other X chromosome that sort of, it covers or hides the recessive form there. So she's considered a carrier though, because these break off in segregation during meiosis, and a male could get that X with a little h there. Okay, and then there's this one, X little h, female, homozygous, hemophiliac. Yeah, there are female hemophiliacs out there, but not many. It's very rare. Now we have XY, that's a male, neither, normal. And then we have X little y, X little hy, the male, neither, hemophiliac. You see, if this carrier was this person's mother, then that's the story. This is the parent, the female would give this to her son. And now the son will be a hemophiliac. Okay, what is the chance of hemophilia in the children of a mother who is a carrier for the disease and a father who is normal in terms of hemophilia? Okay, let's set it up. Just again, like male and female, we have to designate that. All right, so for the daughters, there's 50% would be normal, 50% would be carriers, but there's no hemophiliacs in that group. And then for the males, there's 50% could be normal and 50% could be hemophiliacs. This is again probability. Can the parents described above have any daughters who are hemophiliacs? No. In general, are sex-linked traits seen most often in males or females? Males, because males only have that one chromosome, one X chromosome, so the Y doesn't have any of that. So it's not protected against it, whereas a female could be, and then she'd be a carrier. And color blindness is the same story. It's very similar to hemophilia. You run it by the same way. Multi-gene inheritance, polygenic. Unlike the traits studied by Mendel, many traits in both plants and animals do not appear in two contrasting forms. For example, humans are not either tall or short, really. In human height, that shows a continuous thing going across where there's a range of people from being short to tall. Other examples of traits that show continuous variation are skin color, hair color, eye color, shoe size, hand span, and intelligence. Traits that vary in a continuous manner between extremes are not controlled by the alleles of a single gene, but by alleles of probably multiple genes. And what is multiple gene inheritance? It's any group of genes that together produce a wide range of different phenotypes or physical traits. All right, so that's that. Thanks.